Good morning. In the video we are going to discuss about the second messenger system. Second messenger system is important for the mechanism of action of hormones. So in the endocrinology, this is acted as a signal transduction pathway. This is the basic mechanism to understand the hormonal function. Act as the first messenger. Hormone bind with the receptor. It generates the signal mechanisms, produce the signal amplifications, so it will produces the physiological action. The receptor may be surface receptor or intracellular receptor, whether it may be present in the cytoplasm or nucleus. After utilizing the receptor, it is destroyed by the lysosomal enzyme. It is recycled or reutilized for the another hormone is act through the cell. It is large glycoprotein. It is recycled for the another hormonal action. The quantity and sensitivity of the receptor mainly depend on the concentration of the hormone in the plasma. So when the concentration is less for the long time, it will increases the number of the receptors. That is called as the upregulation. So when the hormone decreases hormone for the long time, it increases the number of the receptor. So this is the upregulation. So it is responsible for the denervation hypersensitivity. The longer time the hormone level is decreased, the receptors in the cell, whether it may be cell membrane or intracellular area, the hormone is secreted, it will respond very high. That is called as the denervation hypersensitivity. When the hormone level is high for the longer time, that will decreases the number of the receptor. That is called as the down regulation. So excess hormone produces the down regulation. So when the hormone level is excess, it will not give the exact action, quick action. That is called as a desensitization. So sometimes the same desensitization occurs for the same hormone or the different type of hormone. That is called as a, when it is response to the same type, it is homologous desensitization. When it is the other hormones, that is called as the heterologous desensitization. G protein is the intracellular protein. Peptide hormones mainly act through the G protein. The G protein connects the receptor with the second messengers. When the hormone binds with the receptor, that receptor is hormone receptor complex, create the second messengers through the G protein. So it converts the signal into the biological activities. The G protein exchanges GDP for GTP. So the GTP protein complexes generate the intracellular signals. G protein has the intrinsic GTPase activity. It after over the hormone functions, the G protein is recycled for the next hormone action. This flow chart explains the action of G protein. The hormone receptor complex activate through the G protein. The G protein alpha subunit. GDP exchange for the production of the GTP. So it produces the intracellular signal transaction. It produces the physiological actions. When the action is over, the G protein has the inherent GTPase activities. Again, the GTP is exchanged for the GDP. So G protein in the rest state for the next hormonal action. Types of G protein. It is divided into two types: small G protein, large G protein. Small G protein again divided into the three subfamily: RAP, RAC, RAS families. RAF family important for the vesicle movement between the cell membrane and cytoplasm. RAC family mediates the interaction between the cytoskeleton and cell membrane. The RAS family is important for the cell signaling from the cell membrane to the nucleus. Large G protein has 5 subfamilies: GS, GI, GT, GQ, G13. The large G protein has 3 subtypes: alpha, beta, gamma subunits. So when the hormone is bind with the receptor, so the alpha subunit is separated from the beta and gamma subunits. So when the alpha subunit is bound to the GDP, it is exchanged for the GDP. So alpha subunit has the intrinsic GDPase activity. It is made to regulate the hormonal G protein signaling. The alpha subunit, alpha S subunit activates the adenyl cyclase. Alpha I subunit inhibit the adenyl cyclase. Alpha Q subunit activate the phospholipase C. So these 3 points are important. Alpha S means stimulation, stimulate adenyl cyclase. I inhibit the adenyl cyclase. Q activate the phospholipase C. You got to remember these 3 points. So see this diagram. So when the hormone is not attached with the receptor, the G protein, all the subunits in the large G protein, alpha, beta, gamma is unite, attached to each other. Okay. So in this diagram, when the hormone is attached with the receptor, the alpha subunit is separated from the beta and gamma subunits. The beta and gamma is united. The alpha is separated from the beta and gamma. It produces the signal transduction, signal amplification. It produces the physiological effects. The hormones, chemicals act through the G protein coupled receptors. Catecholamines like epinephrine, norepinephrine, dopamine, acetylcholine, histamine, serotonin, and adenosine, opioids act through G protein coupled receptors. And angiotensin II antidiuretic hormone, oxytocin, vasoactive intestinal peptide, parathyroid hormone, thyroid releasing hormone also act through G protein coupled receptors. And glycoprotein hormones, TSH, thyroid stimulating hormone, follicle stimulating hormone, FSH, luteinizing hormone, LH, human chorionic hormone, gonadotropins. So many other hormones also act through the G protein. The arachidonic acid derivatives like thromboxane A2 also act through the G protein coupled receptor. Hormone is the first messenger. When the hormone is bind with the receptor, it activates the second messenger system, otherwise called as the signal transduction system. Important second messenger systems are adenyl cyclase, cyclic AMP system, and membrane phospholipase phospholipid system, and guanyl cyclase cyclic GMP system, and transcription of the mRNA. When the hormone is bind with the receptor in the adenyl cyclase cyclic AMP system, when the hormone is bind with the receptor, it activates the G protein, GS or GI. When it is activate the GS, it increases the formation of the cyclic GMP. When it activates the GI, it will decreases the formation of the cyclic AMP by the activation of the enzyme adenyl cyclase. Adenyl cyclase converts the ATP to the cyclic AMP. This cyclic AMP is activate the protein kinase A. Protein kinase A. It produces the phosphorylation of the cellular enzymes, produce the various phosphoproteins. It produces the physiological effects. Same time, the protein kinase produce the phosphorylation of the cyclic AMP responsive element binding protein into the nucleus. It produces the transcription of the genes. It produces the protein synthesis and produces the physiological effect. So this is the adenyl cyclase cyclic AMP system. Termination of the cyclic AMP action by cyclic AMP is converted to the 5' AMP by the phosphodiesterase enzymes. It is also degraded by the serine 309 phosphatase. Dephosphorylation will occur. In the clinical importance in the cholera, it is produced by the Vibrio cholerae. It will increase the cyclic AMP level. It produces the rice water stool. In the pertussis, Bordella pertussis produces the pertussis. It is otherwise called as the whooping cough. In these conditions, the cyclic AMP level is decreases. So in this slide, we are going to see what are the hormones act through the second messenger, cyclic AMP. So the increasing cyclic AMP formation is activate the growth hormone releasing hormone, corticotrophin releasing hormone, gonadotrophin releasing hormone, follicle stimulating hormone, luteinizing hormone, norepinephrine via beta 1 receptors, antidiuretic hormones via V2 receptors. So inhibiting the cyclic AMP formation decreases the cyclic AMP formation act through the norepinephrine via B2 via alpha 2 receptor and somatostatin. Membrane phospholipids phospholipase system via the second messenger inositol triphosphate 1,4,5 triphosphate and diacylglycerol. When the harmone is bind with the receptor, it activates the G protein Q, GQ. It activates the phospholipase C. Phospholipase C degrades the membrane phospholipid triphosphate phospholipid inositol triphosphate to the inositol triphosphate IP3 and diacylglycerol. Diacylglycerol activate the protein kinase C, produce the phosphorylation of the intracellular protein, produce the physiological effect. The inositol triphosphate releases the calcium from the endoplasmic reticulum, bind with the calcium binding protein like calmodulin. It produces the physiological effect. Via IP3 inositol triphosphate and diacylglycerol are norepinephrine acting via alpha 1 receptors, antidiuretic hormone acting via B1 receptors, angiotensin 2, thyrotrophin releasing hormone. These are the hormones acting through the IP3 and diacylglycerol second messenger system. Guanylyl cyclase-cyclic GMP system. So when the receptor is bind, hormone is bind with the receptors, activate the submembrane enzymes guanylyl cyclase. It converts the guanosine triphosphate to the guanosine monophosphate. It activates the cyclic GMP dependent kinases and phosphatase phosphorylation of the intracellular protein, produce the various phosphoproteins and produce the physiological effects. The Guanylyl-cyclic GMP system activates the phosphotransduction pathway in the rods and cones. The phototransduction pathway is occur through the cyclic GMP system. The hormones like atrial natriuretic peptide and nitric oxide act via the cyclic GMP, and enterotoxins of the E. coli also act through the cyclic GMP, and GI hormones, guanylyl also act through the cyclic GMP system. Intracellular receptors. Glucocorticoids receptors are located in the cytoplasm. Thyroxine and steroid hormones 125 dihydroxycholecalciferol and retinoic acid. These receptors are located in the nucleus. The steroid receptors structured as the five domain A, B, C, D, E. A domain is the transactivation domain. A, B domain is the B domain is the C domain is the DNA binding domain. And D is the nuclear localization domain. E is the hormone binding domain. This is the carboxyl terminal. The hormone binding domain is close to the carboxyl terminal. And the cysteine domain is the DNA binding domain. And the DNA is binding domain bind with the DNA with the two zinc fingers. Cellular receptors system. The hormone is enter into the cell, bind with the nuclear receptors in the nuclear receptors near the carboxyl terminal. The DNA binding domain is covered by the hormone responsive element HRE protein or heat shock protein. The steroid hormones, it is covered by the heat shock proteins. It produces the transcription of the mRNA, produces the protein synthesis, produce the physiological effect. Steroid hormone, thyroid hormones mainly act through the nuclear receptor. The nuclear receptor heat shock protein covers the DNA binding domain. In the steroid hormonal action, it is called as the stress protein. It covers the DNA protein. Heat shock protein or hormone response element is called as the stress protein here. Kinase system. So in the tyrosine kinase system, G protein is not activated. When the hormone is bind with the receptor, it activates the tyrosine kinase. The autophosphorylation is occur inside the cell. And the hormone is bind with the receptors. So you activate the tyrosine kinase. So it produces the phosphorylation of the serine and threonine kinase. It produces the enzyme activation and gene transcription, produce the physiological effect. So tyrosine kinase system is insulin is activated mainly through the tyrosine kinase system. Other substances like insulin growth factor 1, insulin growth factor 2, platelet derived growth factor, epidermal growth factor, colony stimulating, monocyte colony stimulating factor. These are the substances act through the tyrosine kinase system. You should not forget insulin is mainly act through the tyrosine kinase system. Growth hormone is acted through the JAK-STAT pathway. When the hormone bind with the receptor, receptor is the dimeric receptor, it activates the JAK kinase, it stimulates the signal transducer activators of transcription, it produces the gene transcription within the nucleus, produces the physiological effects. So growth hormone is mainly act through the JAK-STAT pathway. Growth hormone and prolactin. Prolactin also act through the JAK-STAT pathway. The calcium-calmodulin system, calcium is act as the second messenger. When the hormone is bind with the receptor, it open the ligand-gated calcium channel. Calcium is entered into the cell. It increases the cytoplasmic calcium level. Bind with the calcium binding protein like calmodulin, troponin, call binding. So same time the store operated calcium channel is activated through the inositol triphosphates. So the calcium is act as the second messenger system here. How hormone act on the cell is seen in this slide. Hormones, chemicals opening or closure of the ion channels. Acetylcholine act on the nicotinic receptor, norepinephrine on the potassium channels in the heart. Via adenylyl cyclase, norepinephrine via beta 1 receptor increases the cyclic AMP, norepinephrine via alpha 2 receptor decreases the cyclic AMP. Activation of the phospholipase C through diacylglycerol inositol triphosphate formation. Angiotensin 2 norepinephrine via alpha 1 receptor, ADH via B1 receptor is activated through the IP3 DAG system. Via cyclic AMP, ANP and nitric oxide is activated. Via tyrosine kinase, insulin is activated. Via serine threonine kinase, inhibin activin is activated. Via nuclear receptors, steroid hormone and thyroid hormone is activated. Summary: second messenger system is the basic mechanism of action of the hormone in the endocrinology. Second messengers are cyclic AMP, inositol triphosphate, diacylglycerol, cyclic GMP, guanosine monophosphate, and transcription of the mRNA, calcium, intracellular calcium. These are act as the second messenger. This is act through the various systems we described in this video. So this is the basic topic for the endocrinology. Thank you for watching this video. If you like this video, share your friends. Those who are not subscribed to the channel, please subscribe. Thank you once again.